name is MonicaTschang, and I've been researching potential spinal cord injury therapeutics. One of the biggest obstacles to treating spinal cord injury is the blood-brain barrier, which is a natural boundary that filters what can or can't access the central nervous system. It often blocks things like peptides from crossing into the spinal cord. This is an issue because a lot of current therapeutics found to improve recovery after spinal cord injury are actually peptides. For example, my lab discovered that a peptide sequence derived from the effector domain, or ED, of a protein that we call MARCs can enhance growth of finger-like extensions called neurites of neurons derived from female mice. This peptide also improves the functional recovery after spinal cord injury in female but not male mice. To take advantage of the therapeutic potential of this ED peptide and also circumvent the blood-brain barrier, we found 5 small organic compound alternatives that mimic the structure of the ED peptide by screening libraries of drugs previously used in clinical settings. To now determine if these 5 compounds, which we refer to as mimetics, also mimic the function of this peptide, we asked 2 things. 1, do these mimetics also enhance neurite outgrowth only in neurons derived from female mice? And 2, if so, does it do so through the same mechanism as the peptide? So to answer question 1, we see that in figure 3, yes, actually all 5 mimetics trigger a significant increase in neurite length in neurons from female mice when compared to the control solution. And none of the mimetics affect neurons derived from male mice. And then to answer our second question, we compared the cellular mechanism of the ED peptide to that of the mimetics. So cellular mechanisms or signaling pathways often look like a relay race where a baton is being passed from one runner to another. Previous studies show that treatment with the ED peptide triggers the protein MARCs to get this baton, in this case a phosphate group. So we checked if after treatment with the mimetics, MARCs also has this baton or phosphate group. And the results of this experiment were inconclusive, but generally we didn't see a strong increase in phosphorylated MARCs, so the data suggests that the mimetics activate different runners, or in this case, proteins. In the future, we also hope to investigate the sex-specific response and see what would happen if we added androgen or estrogen inhibitors. And while more work needs to be done to map out the mechanism of the mimetics in order to anticipate potential side effects, if they were to reach a pharmacological stage, these results suggest that the mimetics may provide an efficient treatment to spinal cord injury, neurotrauma, or neurodegeneration at large.